Hey everyone, we're going to continue into chapter 3. Again, our first section of chapter 3, the video focused mostly on amino acids and their structure and names. And here we're going to look more into peptides, their naming and structure, how they ionize, and then we'll get a little bit into proteins and some methods to characterize both peptides and proteins. So first, what is a peptide? A peptide is just a sequence of amino acids that has been condensed together. This is usually a short sequence of about 20 to 100 amino acids. They're relatively small compared to proteins. Again, there isn't a very hard limit on what we call a protein and what we call a peptide. Generally, we decipher it by their size via molecular weight. So how heavy they are based on how many atoms they have in them. Generally, anything less than 10 kilodaltons, we're still going to call a peptide instead of a protein. The way that you make a peptide, again, is just by condensing two amino acids together, specifically at the carboxylic acid end of one amino acid and the amino end of another amino acid. Notice that the OH on the carboxylic acid and one of the hydrogens on the amino group condense together to form water and leave. And in this reaction, a bond is directly created between the carbon on the carboxylic acid and the nitrogen of the amine between two groups. This is called a peptide bond. And again, it's where this condensation of two amino acids has occurred. Amino acids have a variety of functions. They can be things like hormones and pheromones. So for example, a very classic one is insulin. Insulin is a signaling molecule. That's what a hormone is. And specifically, it is a signaling molecule that is a short sequence of amino acids. And so it is a peptide. Oxytocin is also a peptide, a short sequence of amino acids, and is used as a hormone. There is also neuropeptides. Substance P is a specific type of neuropeptide that helps in pain mediation. Certain antibiotics that are very well known are also peptides. Polymyxin B, this is something that we use for gram-negative bacteria. And bacteriocin, which is used for gram-positive bacteria. There's also other types of polypeptides found in nature, aside from examples we've just stated. So things like toxins or that are used for protection in certain animals are also peptides. You can see certain examples like chlorotoxin, which is a toxin used by scorpions for its protection. Now, to go to proteins, proteins again are anything that are larger peptides. They're larger than 10 kilodaltons. They're just a larger sequence of amino acids linked together. But proteins uniquely can have additional groups on them. So when a polypeptide is covalently linked with other items, these form in the whole of a protein. So for example, things called cofactors, which are anything that is not an amino acid component, but is still functional in the whole peptide or protein. These can be things like metal ions or other small organic molecules. Again, non-amino acid organic molecules, but other molecules that are encapsulated in the protein or peptide. Again, these are called cofactors. Co because they're working together. They're factors that are working together for the use of this peptide or protein. There are also things called coenzymes. This is similarly any other functional component that is acting in relation to these polypeptides or proteins in its function. They are usually organic in nature. So they are any cofactor that is organic. We just call them coenzymes. That means that coenzymes are a type of cofactor. An example of this is something like NAD+. This is an organic molecule that we already find circulating in biological organisms, and it is used in tandem with lactate dehydrogenase. There are other examples, prosthetic groups, things like heme, which is a large molecule, and other modifications that comprise proteins and alter their structure in a way that make them kind of a conglomeration of these polypeptides, these peptide chains, and other units.